I'm Farren representing the people who've been involved in the Safety Engineering Working Group. So everything that I'm doing reflects collective thinking work that all of these people have contributed towards. And similar to other talks we've heard earlier today, this really reflects our current thinking or work in progress, and we're very much looking forward to the panel session where we can get some of your thoughts and ideas to further advance our thinking on this problem. Just at a very high level, and this is basically taken from our charter, what we're really been trying to do is to identify unique aspects of the GP Right effort that distinguish this project from other related efforts and establish a framework for investigating and ensuring the safety of all GP Right efforts. And from an operational perspective, we kind of broke this down to sort of three main foci: looking and assessing risk, identifying existing technical safeguards, and trying to project what additional safeguards we might look like in the context of GP Right, and then thinking about safety measures as well as messaging that we should be thinking about with regard to GP Right. Just to give a metaphorical example, there's a lot of precedents, particularly in the internet, that really highlights the significance and need for ensuring safety and security in efforts that we're going to be pursuing in GP Right. What I'm showing you here are basically a whole host of IT companies whose sole focus is aimed at IT and computer security. So you can see that the demands of information and communication that are facilitated by the internet have really required the development of complementary capabilities to secure and safeguard that information. Clearly, this was in the highlight just of the past few weeks, right, where we saw Mark Zuckerberg actually discussing some challenges that Facebook had about securing their information of all of their Facebook users. And so we really want to sort of build off of this type of a work and blueprint by thinking of ways to safeguard the biological systems that we're working on. And ultimately, what we want to avoid is seeing a picture like this in the next 5 to 10 years. So, now that I've got your attention, unfortunately, George was not here to receive that. But I owe him some jokes. For those of you who know, he's played some on me in the past. So you can share that with him over lunch. So what I've also done is presented at least some rationale for safeguarding genetically engineered organisms. And here, I've actually lifted some ideas and concepts from some proposals and grants that I actually wrote with Jeff Buka a number of years ago, where we had a whole program on developing safeguards. And these are a number of reasons we identified that highlight the significance of pursuing safeguards. So obviously, the first is what we often think about is preventing the accidental release of genetically engineered organisms in the environment. Obviously, this type of a idea brings up images of a lot of the challenges that Monsanto has faced over the past few decades. Clearly, there's a need for regulating pathogens, which we're not looking or exploring in the context of GP Right. Other important aspects are protecting the actual scientists who are conducting the work, as well as the environment from highly engineered organisms. From more of a industrial perspective, and sort of building off that last slide I showed you, really securing highly engineered and valuable organisms could be of great utility and benefit to the bioeconomy. And as our ability to engineer organisms really continues to advance, there are growing uses of them in more open types of systems. And so really need to think about developing robust strategies to safeguard those. So what this slide really does, it breaks down how we envision the use of GE organisms. most of which, and over the past 40 years or so, has really been focused on closed or lab-based systems, really with an emphasis on discovery, with, in some cases, particularly in the context of plants, some unknown environmental safety, but most of this is typically safe and well established. You can use certain types of cell lines in the context of plants, for example, tobacco BY2 cells, as well as protoplasts, as some good, safe lab systems. And obviously, there's been great value in engineered systems for bio-waste productions of all sorts of valuable compounds. And as I mentioned just a minute ago, there's this growing use of using these GE organisms in open systems applications. Most of which really we expect have pretty benign phenotypes. And this is also where we want to keep an eye towards using biocontained or bioconfined, which is some terminology that Neal Stewart also introduced from more of the plant biotech world, pollens and seeds, as well as organisms that, for example, have male sterility. So what are some actually real-world needs for safeguards? Well, there's a classic example that actually occurred in the summer of 2009. At this point, I was just upstairs on the second floor working on the first E. coli project when this news hit the headlines. And it was really striking because the biomanufacturing pipelines for 2 of Gensem's orphan drugs were compromised by virus contamination. And I think this really highlights some of the real-world challenges and demands that really speak to and motivate the development of genetic isolation and multivirus resistance, which we heard about this morning. And there are a number of other examples as well. And when you think about deploying GE organisms into more open systems, again, the need to confine them to grow in defined synthetic environments and not in the wild becomes important. That's where intrinsic biocontainment becomes a central focus. I'll also point out, and we heard this earlier this morning, a lot of the first perspectives and thinking of this really stemmed from the 1975 Asilomar meeting during the dawn of recombinant DNA technologies, which really laid out a good framework that we're really building off today. And that's really what established a lot, for example, the physical containment, environmental health and safety structures that are in place at all of our institutions. But really going after the problem of intrinsic biocontainment has been quite challenging. And so from a risk perspective, and sort of bringing this back to GP Wright, we sort of asked the question shown here in the upper right, are there specific activities within GP Wright that require special consideration of risk? And the way that we're thinking about this is we effectively want to start with existing frameworks, right? First established and presented by Asilomar, and then really adopted by our institutions and environmental health and safety offices. And what I'm showing you on the left, basically, you don't have to look at all these details, but it's basically Harvard's version of their Committee in Microbiological Safety. And it just basically shows you a workflow of questions that will basically determine the amount of risk associated with your work. Does it involve recombinant DNA technology or not, et cetera. And so we very much feel, at least the current thinking from this group, is that the first answer to that question that we're posing is really working with existing infrastructures and identifying what might there be any potential additional risk. It will be important to first engage with the institutions where we're conducting this work, and then more broadly, having a mechanism established through some sort of governance across GP Wright that will allow collaborations on the same project and liaising between those efforts as well as these environmental health offices work with us to really address the answers to these questions. And then as the project continues to grow, we basically anticipate ongoing efforts of risk assessment and continued monitoring as we sort of design even more advanced genome redesigns. And the credit for this slide really goes to Mike Chou and Peter Carr, who first tried to basically take a crack at what a GP Wright risk assessment might look like. Again, interfacing with the key stakeholders and scientists and institutions where this work is being conducted, and really starting to develop a perspective on what, if any, of the projects that we're pursuing require some additional surveillance and then possibly technical safeguards to develop. So now transitioning to those elements, I won't go into any of these in detail, but I'll just highlight a few papers that have come out in the literature over the past few years that have developed some important safeguards, and these are primarily microorganisms and bacteria as well as yeast. And you can imagine that a lot of the advances here could really serve as a blueprint to porting over into human as well as plants and other multicellular eukaryotic systems. And of note, the recent work of Pam Silver's lab was actually implemented in the gut of a mouse. So that's actually a good example of applying a safeguard into a open system in the gut of a mouse. So that's, I think, an important step in that direction. And then some work from the plant side, and this is really benefiting from a lot of the insights that Neal Stewart brought to this group. You could think about identifying certain types of plants that would lead these efforts, in particular potato, which does not produce many flowers and seeds, and it's mainly vegetative. And so therefore it could be a good chassis organism that could serve as a natural bioconfinement or biocontainment system for genetically engineered efforts. And so going back to one of the central foci that we spoke about at the first panel this morning, and the real emphasis towards recoding and development of ultra-safe cell lines, right at the heart of that is recoding. And what I think is important to point out here is that the actual act of recoding naturally confers important safeguards. In particular, genetic isolation and multivirus resistance, which we've published a few papers demonstrating proof of concept on this in E. coli, which I think further inspires additional recoding work that you heard briefly from George's lab, as well as using that as a blueprint in other species, which we spoke about this morning, and then effectively taking codons that we've been able to remove, reintroducing them, and actually engineering these recoded organisms such that they depend on synthetic amino acids for viability. So you can imagine that you can see recoding can basically address important problems on isolation or virus resistance, as well as biocontainment, by requiring them to grow in the presence of a synthetic amino acid, which does not exist in nature. And the last thing, and I think the cool extra benefit that we can extract from these recoded organisms, is you can also repurpose those open codons and start to site-specifically encode lots of new types of amino acids or even modified backbone monomers into proteins and polymers, and really use this as a chassis to produce entirely new types of biologics and therapeutics. So I think there's a lot of really compelling reasons, both on the safety side as well as the biological functional side, that I think make the pursuit of recoding an exciting and central focus of this effort. So now touching on some safety aspects, we thought it would be important for this group to think about safety both in terms of technical safety in the lab or in the field, as well as how we communicate this and develop outreach efforts to the public. And here, as this quote by George Shaw sort of nicely articulates, it's really important to be transparent and candid in everything that we're doing, which is obviously a major focus of our efforts today and throughout this project. And I'll lastly just include this quote that I often refer back to from the Presidential Bioethics Commission that was published at the end of 2010. I think it's still relevant today, in particular to the efforts that we're pursuing here, where there's tremendous opportunity and excitement from a technological and societal impact that we can make with this type of a project. And it's also always important to keep our eye on making sure we're pursuing this by assessing risk and, as the quote here says, being responsible stewards along the way. So with that, once again, thank all the members of the group for some really great conversations. I'd also like to thank the leadership who really have paved the way for this project, and it's been exciting to be involved. In particular, thank Nancy and Amy, who many of you have interacted with over the past year or so, that have kept us on check to make sure that we don't slack too far behind in our group efforts. And also, a few weeks ago, we got some input from Carol and Jonathan from the LC group that was helpful in some of our thinking surrounding risk. And we welcome new members with diverse backgrounds and expertise to participate in our working group, as well as more interactions with other working groups, where I think there's lots of synergies that we're seeing emerge, and I think it'd be good to use this meeting as an opportunity to launch that as we go forward. So with that, I think we have one of the few groups where all members are present today. So I'm going to ask everyone to come up, grab a seat, and we'll shift now to the panel discussion. While Pete, Mike, Daisuke, Brad, and Neal grab a chair, I think they will occupy all those chairs. So I'll just hang out here. I'll just throw up some questions that we sort of circulated internally that I thought would be good starting points for our discussion. So while they're sitting down, you guys can take a look at some of those questions. And what we'll do is, starting with Daisuke, we'll just ask you guys to quickly introduce yourselves and a little bit about your background and your interests in GP Wright and this particular working group.
Thank you. I'm a mic on?
Just start talking a little more.
I'm Daisuke Kiga from Japan, Waseda University. My main work is another recording of genetic code. I have removed some amino acids from genetic code, but this is in vitro one. And I also enjoy IGM activity more than 10 years. That's why I'm in here for safeguarding. Thank you. And so my role in this working group is introduce Japanese situation for synthetic biology and GP Wright, and also biosafety and biosecurity. Because I have a collaboration with Japanese biosafety and biosecurity people. Thank you.
I'm Brad Schmier. I'm a research fellow at Sloan Kettering. I work on interested in problems in DNA and RNA repair, genome assembly, and I also have had a long interest in scientific education and communication and how these are communicated, how synthetic biology and GP Wright communicates to the public. So,
hi, I'm Mike Chou. I work on the Personal Genome Project with George Church. I also got my PhD with George, and my rotation project was doing synthetic biology. And one of the reasons I stopped doing that is because I was concerned about some of the safety issues that we hadn't addressed. So I guess my interest in this is just, I've been interested in safety for a long time of synthetic biology.
So I'm Peter Carr from MIT's Lincoln Laboratory, where I lead a synthetic biology research program, and our focus is primarily on technology development, but it also intersects with a number of questions about potential ways that synthetic biology and related technologies could potentially be used intentionally to do harm, or accidental harms that could arise from the new technologies. And a lot of my interest in particularly in this group includes both the engineering side. I had a great opportunity to collaborate with George and Farren and others on the first E. coli strain, the 63 codon strain, but also then the questions. I really hope to bring greater granularity and resolution to the questions we ask about what are potential risks, and specifically what kinds of harms are we actually concerned about, to look at that in greater detail so that we can have a better understanding of when something genuinely is a concern versus, when you get past the hand-waving level, maybe certain concerns actually start to evaporate.
Yeah, my name's Neal, Stewart, University of Tennessee, also known as the plant guy. And after working in the area of plant biotechnology for the past 25 years, I'm pretty motivated to help this emerging field avoid the GMO controversy and all the baggage that went with that, and that we still experience, even though there have been somewhere around 100 trillion genetically engineered plants in the field in the past 25 years, and no one's died. It's still controversial.
Okay, so what I thought we would start off by just opening up to questions from all of you. Obviously, you see various questions that we've thought of internally, but I thought it would be a good opportunity for some of you guys to introduce some of your own thoughts or questions at this point before we introduce our own bias into this topic. Okay, up right here we've got one person.
So one of the issues with genetically modified plants, for example, has always been about horizontal transfer and things like that, and mating with wild plants, and so is this something that you've thought about in terms of genetically modified animals, so that the idea of accidental release becomes less of a problem if they are unable to mate in the wild? And so have you considered like ways of making kind of conditional mating strains that would kind of obviate that entire potential problem?
I'm not the animal guy, but with plants, yes.
Yeah, as far as I know, I don't think that there are goals of actually creating GP right animals right now. So I don't think that is something that's on the radar screen. Is that consistent with your thinking, Jeff? Is there a microphone? Right over here.
I mean, we've certainly talked about doing mice. So I think the whole point would be that we would want to be able to propagate them in the laboratory, but making such, germline transmission somehow conditional would definitely be an interesting thing to consider, because mice do have a tendency to get away.
So one of the things that we do in the boring part of IT is we run tabletop exercises where, frequently an outside consulting group will give us a scenario, and then you get the stakeholders in a room and run through sort of on paper what your plan was and what actually happened. Is GP right to the point where we might be able to do a similar sort of exercise, or is it just way too premature because we're not even to the point where we have systems going out?
I don't think that is too premature. I think that's, in fact, a good idea. And our initial conversations have had a certain element of that thought process in them. And one of the things that we really want to balance, and it really highlights our need and desire to work with some of the other subgroups, like the LC subgroup and the communications group, is trying to balance maximizing transparency and, of course, maximizing responsibility. And at the same time, that requires being free to speculate on potential harms that may be of low probability and may be real or may be essentially unreal. But to be able to decide and discuss that whole range, and at the same time to be able to communicate the results of that conversation in a way that does not induce a disproportionate degree of fear. And that, I don't think that's a solved problem, but that's part of the problem we want to pursue.
I'd just like to second that and add, so Neal said how many organisms we made that we haven't had a problem yet. I mean, we as scientists implicitly assign probabilities to events. Things like the Large Hadron Collider, remember when they were going to spin it up, people were like, well, you could create a black hole. Well, and then scientists would say, well, no, there's no possible way we're going to create a black hole. And then somebody else would say, well, why are you doing it? And they said, well, we got to do the experiment to see what happens. And so scientists make educated guesses about what the probability of things that are happening. And I think it's important when we communicate to the general public, as Pete said, what is a possible chance that something would happen? We have to explore these things, and then we kind of sort of step back and say, well, how will we put a safeguard in to make sure that doesn't happen, or that what is the probability of that? With the example with the setting off the first atomic bombs, there was this.
The story from World War II that had evaluating the question of the chance of a bomb setting the entire atmosphere on fire.
And we decided to do it anyway as a society somehow. I'm not sure any of us if we're old enough to be there, we're aware that that was being a decision made for us. But I think that so the part of the conversation of the public has to be somehow we have to communicate these probabilities. It's a difficult thing, but that's why we try to engage the LC and the communications group with our discussions, and we really want to engage the other groups in the same way.
Just add to that with the IT also, that the design plans, that the sequences, that how does that come into safety? I think that'll be, the recipe is.
Right back there.
Thank you. A lot of the discussion tends to be around how the professional community, if you will deal with safety, policy, and procedure and such. But as we look at technology evolving, the barrier to entry tends to get lower and lower. We're talking here about communicating with the public so they understand the risk, but over the next 3, 5, 7 years, the public will actually become the risk as they start getting access to new technologies, new resources. It's all fine and well for us to develop policies and procedures to maintain safety, but when every high school student is also going to be able to do the equivalent of what we in a major institution do today, how do you approach that from a safety point of view? It's not just telling them we're safe, they're now becoming a bigger risk than we ever will be.
I think that's a meaningful question, as regardless of whether we might argue over specifics and timeframes, the barriers are coming down, and people are getting more access to those technologies. And I don't think of it as much as the public becoming the risk as the public joining us in considering and being a part of the whole risk consideration landscape. I remember back when it was an incredibly novel thing to run a gel visualizing DNA in high school, and now that's relatively commonplace. We see access to DNA synthesis, and of course this whole project depends intimately on advances in DNA synthesis. I certainly don't have any easy answers. I think the education along the lines of risk has to go hand in hand with the education, with the technology. I didn't spot some of my colleagues from the community labs here yet, but they've done an incredibly laudable job of demonstrating. Thank you. Oh, sorry, I didn't see you earlier. They've done a very laudable job of education on safety and larger issues that as an academic I'm sort of jealous of, because it's better than I received, if you will, in my own training. So that gives me encouragement. It doesn't solve the problem, but it gives me a lot of encouragement.
I would also add that, the emphasis that we're putting on here on safety and LC and other aspects that go beyond just the scientific goals, I think speak to the importance that we put on these issues. And as you can see, a number of us have actively developed technical safeguards. And we are also, as you can see, asking questions, what, if any, of the projects that we're looking to pursue require the implementation of these? And so we're establishing a discourse on this. We're developing a culture that establishes the significance of these aspects, and we're actually implementing them in our efforts. So I think it sets the right model as these types of technologies start to percolate more broadly.
So may I add one phrase in Japan? We have a phrase of anzen anshin. Anzen is safety, but anshin is a feeling without fear. So for engineering of a scientific point of view, we can multiply probability and outcome of disaster. But in the case of anshin or some feeling without fear, general public, or even for me, tend to add probability and outcome. So and I also have fear for something I don't know well. That's why we need risk, not only safety communications, especially for why GP Light can do something to decrease, how to say, disaster or probability of disaster. Thank you.
I think we've got time for one more question. Over here.
What is the threat of a synthetic human chromosome? Or what is the threat of a synthetic human chromosome in a cell or in a person? I mean, what is the specific worry that you would have about it?
So, could you repeat the question?
What is the threat of a human chromosome, right? What is the threat of it?
What is the specific risk?
I'm not scared of it yet, so let me know what I should be worried about.
Yeah, I think one of the things we talked about is not just the product of what we do, but the processes that we develop. So the end product of this project is not going to be just a synthetic genome or a synthetic organism, but actually how we go about doing that. So the end product is not just a specific organism you can contain. It will be technologies and processes that get there. And so those things may also pose potential risk. So I think that the technology we develop, and suppose we develop some new recombinant technology or a new viral vector to build large genomes, what is going to be the risk of that? I'm always amazed at my colleagues that work on viruses on human cells, and what are the safeguards about that? But we've established these kind of parameters for work in our labs today. If we establish new technologies, I think that's what we want to watch for. What are we doing that's different in this project? To make a huge genome or a chromosome, is there something new we're going to have to, fundamentally new, we're going to have to work on to get there? And those are things we want to watch out for.
Yeah, I mean, I would say that, once again, from the past 25 years, people are concerned about the exposure, that something is getting out in nature and will change nature. Okay, and you complicate that, and it doesn't matter if there's no hazard, right? If it's just that getting out. Second thing is who's controlling the technology, and people are a little wary about the big ag companies or any big companies, And then the third thing is really the communication, and so I'm really, really happy we have these working groups, right at the ground floor to discuss this and to show everybody that we think this is really important to do.
Okay.
Thank you.
With that, thanks for all of your time. Happy to interact more during the lunch break.